Dr. K. Rajshekar. So in this session, we are going to go through prelims questions that have come in 2021 UPSC exam and science and technology questions that we are going to focus on in this session. Okay. So how many questions have come from science and technology part? Like, I think it went with the trend that we have been seeing for the last few years. There were totally 11 questions that have come from sciences. But what has changed from last few years is we see more weightage from bio and biotechnology part, applications from biology, biotechnology, which was kind of what we have been seeing for the last couple of years, more weightage from biology. But we also used to see questions from ICT, information and communication technology, and which we have not seen this year. 0 questions from information and communication technology. Okay, more on general science part that we have seen, like 3 questions from physics, 3 questions from chemistry have come. Okay, and no questions at all from space and defense area, and no questions from nano, nuclear, other 2 areas where once in a while we also see questions. 1 question from nano or 1 question from nuclear. Sometimes we have not even seen that this time. So slightly, I think, as you notice, is more focused towards the general sciences and of course applications and day-to-day things that we will notice that in the type of questions they have asked. Okay, right. So let's look into the first questions that have come from the biological area and applications of biology, that is biotechnology area. So first question, in the context of diseases, in the context of hereditary diseases, consider the following statements. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy. The key word here is mitochondrial replacement therapy, either before or after in vitro fertilization of egg. Okay, this question had come in the past as well, related to a specific technique called pronuclear transfer. In a way, they asked again another question related to the same technique. Okay, so what they are saying here, mitochondrial diseases will go from parent to child, and these can be prevented using the technique called mitochondrial replacement therapy. But specifically what they are asking here is, can it be done before or even after fertilization of egg? Meaning sperm and egg fusion, which is what is called fertilization. Can it be done? Can this treatment can be done before and even after fertilization? Question mark. A child inherits mitochondrial diseases entirely from mother and not from father. Is it true that mitochondrial diseases are inherited from mother, not from father? Okay, let's see. So which of the following options are right? So this question, if you really see in depth with technical knowledge, then the answer may be slightly different. Okay, that is a confusion sometimes we have with respect to questions, especially from science and technology, because huge field, lot of developments are happening. What is right or what was right few months ago or a year ago may not be right today. But at least we'll try to see that what probably the examiner here is trying to look into. So I think they were talking about mitochondrial replacement therapy. Specifically, they're talking about pronuclear transfer. If you're focusing on that method, so what normally happens in pronuclear transfer is you notice that they take they take the egg cell from the mother, okay, with abnormal mitochondria. And then what do? They inject sperms, which is process of fertilization. Similarly, they take cell from a donor, one who is carrying no mutations in mitochondria. Okay, mitochondria lies in the cytoplasm outside the nucleus, right? So they took a donor cell where no mutations in mitochondria, at least the mutation that they want to avoid. And then patient's egg cell, okay, that they have taken and injected sperms in both. Basically, fertilization would take place after this. But just before complete fusion, we will have what is called a pronuclear stage. Okay, this 2, like egg and sperm, is just about to fuse. That stage is called pronuclear stage. This method is called pronuclear transfer method. So what do? Once this fertilization is happening, they in the pronuclear stage, they would actually remove the faulty nucleus, mother's and father's nucleus. And what do is they basically place it into donor's cell. So, but they don't take the nucleus of the donor. The blue color nucleus coming from the donor is taken out. What they are placing here is the fertilized zygote from patient. But and in the process, what is happening here? The background is changing, meaning cytoplasm is changing. Cytoplasm should contain mitochondria. The faulty mitochondria would be taken off. Okay, that is what happens here. So here, what you notice is it is happening after fertilization process. Fertilization is done first, and then they added the, or made the change, or conducted mitochondrial replacement process. So pro-nuclear transfer technique approach of MRT administered after fertilization. If they are aiming at or restricting their statement to the pro-nuclear transfer technique, then the first statement cannot be right because it has to happen only after fertilization of the egg. But if you really look deeper, there are other methods like spindle transfer, etc. Those can happen before or after. Okay, but I'm believing that they are probably focusing on the pro-nuclear transfer technique. Okay, then this statement may not be right. Then second, a child inherits mitochondrial diseases entirely from mother. As far as our understanding goes, I think something is also changing on this side. As far as our understanding goes, mitochondrial diseases are known as maternal, or they are maternally inherited from mother to children. Okay, cytoplasm usually comes from the mother, and the nucleus comes from both father and mother. So because mitochondria exists in the cytoplasm, so what we notice here is that diseases probably transfer from mother to the child. So this statement is correct. So what would be the answer here in that case? So it is only 2 only, or this should be the right answer. Okay, one could be wrong. This is about the first question that we would like to discuss here. Next, consider the following. Straightforward and an easy question. And we have been seeing a lot of applications related to that in day-to-day in the newspapers. So it is not complicated at all. But what is required here is knowing bacteria, fungi, virus. Okay, whether they're asking which of the above can be cultured in artificial or synthetic medium. Out of these three, which of them can be grown in artificial medium or synthetic medium? What is an artificial medium or synthetic medium? Synthetic medium is a medium that you can make by adding different components, like little amount of sugar, little amount of nitrogen source, phosphorus, or other micro elements or macro elements that are needed. Mixing it, if you can create a medium, that is called synthetic medium. Okay, what can be grown on such a medium? It is very well known since long that bacteria can be grown like that. If you see in this image, these tiny dots are called bacterial colonies. Okay, you can grow bacteria like that on the synthetic medium. This has been, done for a long time. Okay, so this is true. Even if you go to laboratories for testing, urine analysis, sometimes they ask for cultures. They're able to culture the bacteria that you may have in your urine, checking for the cause for infection, urinary infection. Okay, so yes, bacteria is definitely can be grown. Then what about fungi? Fungi is also grown. If you see on the right hand side of this image, what you see here is the fungus that we see, mold that we see, can also be grown in the defined medium. So fungus is also used, or fungus is also grown in the laboratory. So what about virus? Can we grow it? Can we grow the virus in the laboratory? Okay, can we grow them on the synthetic medium? No, it is not possible. Think about COVID-19. If it was easy to culture virus, COVID virus would have been nicely cultured, and we would have understood about COVID virus, much quicker than what we are understanding today. So because virus is strictly parasitic, it has to survive inside a cell because it doesn't have its own metabolic action or activities. So here in bacteria and fungus, you are feeding, you are providing food source, it is digesting it, utilizing it and metabolizing it. Virus cannot do. Virus entirely depends on us for this process, or any host for that matter. So virus, it is not possible. So only 1 and 2 can be grown in the synthetic medium, but not the virus. So this is something that indirectly we have been told many times in the newspapers. Okay, but fundamental, very fundamental concept in biology. So option A is the right answer to this. Then consider the following statements, again about viruses. So questions on infections and infectious agents is not uncommon. We have been seeing that this is a very, very important area. So previous question and this question can be connected to health and infections, okay, related to humans. Okay, here what are they asking me about? Adenoviruses have single-stranded DNA genomes, whereas retroviruses have double-stranded DNA genomes. So viruses can be classified into RNA viruses, DNA viruses, depending on what genetic material they carry. Is it true that adenoviruses have single-stranded DNA? Is it true that retroviruses have double-stranded DNA? We will come to the answer. Second statement, common cold is sometimes caused by an adenovirus, whereas AIDS is caused by retrovirus. So what genetic material these two adeno and retroviruses contain, whether and what infections do they cause is what they are aiming at. Okay, first look into the statement one. Adenoviruses, what do they have? Adenoviruses have double-stranded DNA viruses. Look at this image, this is an adenovirus. We have two-stranded, very much like us. Okay, double-stranded DNA is present in them, not single-stranded DNA. If you know this, okay, then you know first statement is wrong. But we may not know adenoviruses as much as we do about retroviruses, because HIV is a retrovirus. We frequently hear about retrovirus and HIV. So, and HIV is not a double-stranded DNA virus. Not at all is a DNA virus. It's an RNA virus, single-stranded RNA. There are two pieces of RNA in HIV. So it's an RNA virus. Even if you know one of the statement, even half of the statement, you are through. So this statement is wrong. It's not right. Then common cold. Look at the statement. Adenoviruses are not very well known for causing common cold, but sometimes they can cause symptoms like runny nose, for example, or itchiness in the throat, etc., or pneumonia. Those things can also be caused by adenovirus. So common cold is sometimes caused by adenovirus. This is true. Okay, otherwise rhinoviruses we talked about, or influenza, those type of things are known to cause these problems. But sometimes adenovirus can cause this problem. And actually, look into the second half of the statement. AIDS is caused by retrovirus. HIV is a retrovirus, so AIDS is definitely caused by it. So second statement is correct, but not the first statement. So which of the above are correct? Only statement 2. So option B is the right answer to this. So very fundamental. These questions we have seen so far are very fundamental. Next, the term ACE2 is talked about in the context of, this is something, a piece of cake for many, because we hear almost every day about ACE2 related to COVID-19 virus. Okay, the spike proteins that we normally hear about, spike proteins, okay, they interact with ACE2 receptor. The spike protein of the COVID-19 or SARS-CoV-2 virus binds to ACE2 receptors on our tissues. Okay, so basically the ACE2 was heard not related to genes introduced in the genetically modified plants, not related to satellite navigation system, not related to radio collar for wildlife tracking. It is spread of viral diseases like COVID-19 viruses. So right answer is the option D. Next, with reference to recent developments regarding recombinant vector vaccines, consider the following statements. Okay, so again, related to COVID-19, not directly, but indirectly, and not limited to COVID-19 infection, because we do use recombinant vector vaccines for many infectious problems. Okay, let's look into the statements here. Genetic engineering is applied in the development of these vaccines. Recombinant vaccines, are they products, are they applications of genetic engineering process? Next, bacteria and viruses are used as vectors. Can we use both viruses and as well as bacteria as vectors in making those vaccines? Let's see. So COVID-19, we have seen many vaccines in Indian market. We have seen Covishield. Covishield indeed is a viral vector vaccine. How are viral vector vaccines made? So if you notice COVID-19 virus, we can take a piece of this genetic material of the virus. COVID-19 is also RNA virus. You pick up a piece of that and place it into an unrelated harmless vector. Vector is a carrier. Okay, so they place this red color DNA portion into it, which actually codes for genetic material for, spike protein, for example. Okay, Covishield, that is how Covishield was made. So when you introduce this virus, which is not harmful to us, carrying COVID-19 piece of genetic information, placed into our cells, placed into human cells, what happens? They grow. As a virus grows, virus multiplies. In the process, they produce spike proteins. And our immune system can see those spike proteins and make antibodies against them. Okay, again, cell-mediated immunity is also possible, but at least antibodies are made here. So basically like that, what happens, viral vector vaccines are possible. We have already seen, we have been using them already. And can bacteria be used? Because viruses are definitely potential, or we have been using them. What about bacteria? Yes, there are many experimental vaccines that have been created where bacteria, like plasmids, bacteria carry extra chromosomal material called plasmids. So those type of plasmids are very good carriers. For bacterial infections, yes, bacterial plasmids can also be used in the same way as we talked about to express the particular gene, so that an antigen can be made in the process and our body can recognize that, and no infection can happen because you're only introducing small portions. That is the benefit of modern vaccines and biotechnology. And using genetic engineering, we can make these more safer, effective vaccines for us. Okay, so genetic engineering is definitely used to make these vaccines, especially to identify and delete genes which are called virulence genes, genes that cause infection in us. We are removing them. We're only keeping 1 or 2 such genes so that our immune system can be trained based on. Okay, and so they're safer because when you remove those virulent genes, this virus cannot revert back to the virulent one or infection causing one. So it is safer. At the same time, you are training your immune system. It is acting like a vaccine. So both statements are correct. Genetic engineering is indeed used in making these vaccines, and both bacteria, viruses can be used as vectors. Okay, so both 1 and 2 are correct for the statement. Okay, right. In this video, I wanted to focus on this biology, and biotechnology part of questions. The next video, I'm going to focus on questions from the physics part and chemistry part. 3 questions from physics have come, 3 from chemistry. But if you notice it, this paper, these questions from science and technology part are very fundamental. Okay, very much expected, may not be directly, but indirectly. So keep in mind when you are preparing for 2020 prelims, keep that in mind that such holistic approach in preparing towards science and technology is very, very important. Conceptually, starting with NCERTs to looking into what type of applications are coming in. Hope this discussion is useful to you. I'll come up with more analysis on remaining questions from science and technology. Thank you, guys.